in order to perform its function, the leukocytes must leave the circulation, the leukocytes must leave their comfort zone and emigrate into the tissues where there is infection. Right? Eta kalke ami bolechilam. The leukocytes must come out, they must leave, they must exit the circulation and they have to go towards that site where there is infection or inflammation, where they are supposed to perform their function. Okay. So the first thing you must remember is leukocyte recruitment to the site of injury. Leukocyte recruitment to the site of injury. Okay. এখানে কি হয়? We need to know three things. You must remember three things. Okay. Margination, rolling, and adhesion. দেখতে পাচ্ছ লাস্টে? ওখান থেকে লাস্টে দেখা যাচ্ছে margination, rolling, adhesion, MAR, MRA, এই তিনটে জিনিস মাথায় রাখতে হবে। Okay. So normally in a vessel, normally in a blood vessel, if we consider this as a blood vessel, the red cells, RRR stands for red cells. These red cells, they have a central location or central axial column. Red blood cells gulo blood vessel এর মাঝখানে থাকে, exactly in the centers. The leukocytes are located towards the periphery. So this is the normal central axial column of blood. Red cells are located in the center, while the leukocytes, they assume a little peripheral position. Normally, this is normal. In case of inflammation, red cells so ekhanei roilo. Inflammation e ki hocche? First and foremost thing is, what happens in inflammation? Blood er ki hoy? What happens to the blood? Yes? The blood flow slows down. Stasis. Right? Inflammation e ki hocche? Stasis hocche. So the wall shear force decreases. Decreasing wall shear force. So what happens is, these leukocytes are already located towards the periphery. They assume a more peripheral position. They move more towards the endothelium, right? They move more towards the endothelium. So this process is called margination. This entire thing is called margination. Eikhane lekha ache margination. Okay. See this picture, normal, and they are moving towards the periphery. Eta the schematic picture. To ami ekhane বোঝানোর চেষ্টা করছি। The red cells are located in the center. In normal, the red cells are located in the center, while the leukocytes assume a little more peripheral position. In inflammation, during inflammation, the wall shear force During inflammation, first and foremost thing is there is stasis of the blood, means the blood flow slows down and the wall shear force decreases, which forces the leukocytes to assume an even more peripheral position, more towards the endothelium. So this particular thing is called margination. After margination comes rolling. Rolling e ki hoy? This is basically a natural mechanism. Why leukocytes assume a peripheral position towards the endothelium? Why? Ami question to tomai korchi. Think logically and answer why the leukocytes move towards periphery, towards the endothelium. Tar karon ki? Huh? Yes, exactly. They basically have to exit the circulation. And to exit the circulation, they must cross the leukocyte, they must cross the endothelium, they must pierce the basement membrane and exit the circulation. So, so that is the reason why they move more towards the endothelium. Okay? So this thing is called margination. Now, rolling e ki hoy, the leukocyte rolls over the endothelium. Seta ami details e aschi. Rolling basically means the leukocyte attaches to the endothelium. The leukocyte attaches to the endothelium and again detaches from it. What attach korche? And after attachment, because of the flow of blood, they again detach from the endothelium. Attach korche, detach korche. So this cycle continues. During this process, the blood flow even slows down. And this attachment and detachment is mediated by a family of proteins called complementary adhesion molecules. Complementary adhesion molecules. Aschiam, details aschiota. Now the blood flow slows down. This helps the leukocytes adhere more firmly to the endothelium. So CAM, complementary adhesion molecules, selectins through te lagche. Kintu that is not enough. Lagche abar beri jache. They are attaching to the endothelium and they are getting detached from the endothelium. So this thing is called rolling. This thing is called rolling. Adhesion ta jodio ache, adhesion ta ki ami aschi ekhane. So this is mediated, this rolling is mediated by selectins, by a family of proteins called selectins. And finally, before they exit the circulation, they must cross the endothelium. Na? So, we lose attachment korle cholbe na. They must attach firmly to the endothelium as a prelude to exit circulation. And this firm attachment is mediated by another family of CAMs, means complementary adhesion molecules, that is known as integrins. That is known as integrins. So remember these names, selectins and integrins. And then the leukocytes exit the circulation through the endothelial spaces. Here's the basement membranes and they exit the circulation. Right, so is this clear? How does normal axial flow looks like and what happens in inflammation? Margination and rolling. Margination, rolling and adhesion ki hocche tale ami bole diyechi. So you know what is margination, what is normal, how does the normal axial flow, normal central column of blood looks like, what is the position of the red blood cells and the white blood cells. Red blood cells are in the center, white blood cells are in the assume a peripheral position. In inflammation, because of stasis, the leukocytes assume an even more peripheral position, move more towards the endothelium. The blood slows down there in stasis. Right? So this is called margination. Then comes the process of rolling. The leukocyte attaches itself to the endothelium and detaches itself because of the flow of blood. Attach korche, detach korche, attach korche, detach korche. This cycle continues and this is mediated by a family of protein called CAMs. I'm coming to it. And which CAMs do it? Selectins. During the process, the blood, so this thing is called rolling. This is called rolling. Finally, the leukocytes come to rest. They attach firmly to the endothelium as a prelude to exit circulation. They have to attach with slow, loose attachment. The leukocytes, the prerequisite to exit circulation is to attach to the endothelium firmly. And this firm adhesion to the endothelium is mediated by another family of CAMs that is known as integrins. Right? So that is adhesion. Clear? এই তিনটে জিনিস কিন্তু মাথায় রাখতে হবে। যদি পরীক্ষা আসে এইভাবে লিখবে, margination, rolling, adhesion, MRA এইভাবে কিন্তু লিখতে হবে। ওকে? সো আমি এটা So adherence কারা করাচ্ছে? CAMs, complementary adhesion molecule, also known as CAM. CAM এর দু রকমের হয়। One is selectin, the other is integrins. Selectin আবার কয় রকমের হয়? L-selectin present on leukocytes, E-selectin located on endothelial cells, and the next one is P-selectin located in platelet and endothelial cells. Selectins কি করায়? এটা হচ্ছে weak attachment. Integrins firm attachment. Right? Okay. LEP means LEP is basically, L stands for leukocyte, L stands for leukocytes, which is located mostly on neutrophils, macrophages. E for endothelium and P, platelet and endothelium. Okay, এই জিনিসটা মাথায় রাখতে হবে। So this selectins, they are involved in weak interactions. After that, they bind to integrins. Integrins are associated with firm adhesion, and everything, these leukocytes, all these leukocytes, endothelial cells, platelets, they all also express ligands for integrins also, ligands for selectins also. Just মাথায় রাখো। Okay, is this clear? এই পয়েন্টটা ক্লিয়ার? আমি ছবিটা এলেই বুঝতে পারবে, এই ছবিটা এলেই তোমরা বুঝতে পারবে। I am coming to that, but before that, what stimulates inflammation? Any kind of injurious agents stimulates cytokines. Any kind of injurious events stimulate cytokines like interleukin 1, interleukin-8, TNF, tumor necrosis factor, platelet activating factor, histamines, components of the complement system like C5a, products of arachidonic acid metabolism. এই টপিকটা তোমাদের নেওয়া হবে মানডে, এই টপিকটা তোমাদের ডিটেইলসে মানডে নেওয়া হবে, জাস্ট রিমেম্বার দ্য নেমস, ওকে, thrombin, etc., etc. So these are all the agents which stimulates the leukocytes, okay, right? So you can remember this picture, not that important because new নতুন রবিনসের যে এডিশনটা আছে, they have deleted this picture. I'm not explaining too much. This is nothing but these are all the p-selectins which are actually located inside the endothelium. Eta explain korche in a normal state when the leukocyte is inactivated or when there is no inflammation, or sorry, the endothelium is in an inactive state, the selectins are located inside the endothelial cells. This is known as Weber palette bodies. Whenever there is any, see, histamine, thrombin, what are they? These are cytokines which activates the endothelial cells. So there has been some kind of inflammation, the endothelial cells has been activated. Then what happens is these Weber palette bodies or the p-selectins which are located inside the endothelium, they move up and express themselves on the surface. They express themselves on the surface. Neutrophils, these green colored things, so this is basically this thing, this yellow colored things, they are the complementary adhesion molecules. P-selectin. Now neutrophils express ligands for this green colored triangular thing which you're seeing like a cone is called as a ligand. This is known as a ligand. Neutrophils express ligands for p-selectins. Neutrophils come and attach to the endothelium. Ami ki bolechilam? Why the neutrophils assume a peripheral position? Because they have to exit the circulation. So before that, they must bind to the endothelium. The endothelial cells express E-selectins or P-selectins, whatever it may be. The neutrophils, they have ligands for P-selectin or E-selectin, whichever selectin has been expressed, and neutrophils bind to the endothelium. কি হলো এটা? রোলিং হলো। Now এই যে সেলেক্টিনের সঙ্গে বাইন্ড করছে, আমি আগেই বলেছিলাম রোলিং এ কি বলেছিলাম, what did I say? These interactions are weak, right? These interactions are weak. There must be firm integration. The integration now must be very firm. ঠিক আছে, এক্সিট করতে গেলে তোমার ফার্মলি বাইন্ড করতে হবে, রাইট? পরে এই পিকচারটা কি দেখাচ্ছে, ইনক্রিজড অ্যাভিডিটি অফ ইন্টিগ্রিনস। এরপরে the leukocytes bind with integrins. These integrins help the leukocyte bind to endothelium more avidly. So this is basically leukocyte adherence to endothelium. This is basically leukocyte adherence to endothelium. Leukocyte by লেগে গেল। So leukocyte এত তোমার পরীক্ষায় লিখতে হবে না, even if you produce 10-15% of what I said, more than enough. কিন্তু নামগুলো মনে রেখো, just don't write leukocyte adherence, anybody can do that. Mention CAMs, complementary adhesion molecules. Complementary adhesion molecules এর মধ্যে explain selectins, tell them selectins, what are the types of selectins, P-selectin, E-selectin, L-selectin, and then say that selectins are involved in weak interactions. For firm interaction, it must bind to, for firm interaction, it must bind to, nice one. For firm interaction, it must bind with integrins. Integrins are involved in firm interaction. So explain this way and highlight them. Okay, it's easier said than done. আমি বানছি তোমাদের স্টেজে আমারও অত ইজিলি আমি পারতাম না, বাট ইউ হ্যাভ টু put effort, that's all. Okay, so this is basically the entire this bird's eye view. This is how it happens. This is how it happens. Okay, this is exactly how it happens. The leukocyte, they assume a peripheral position, they are in the circulation, they are assuming a peripheral position. After assuming a peripheral position, they roll over with the help of integrins, then they bind more efficiently, they roll over with the help of selectins on the endothelium, and then they bind with integrins, which helps in firm adhesion of the leukocytes to the endothelium, and finally the leukocytes exit through the interendothelial spaces, pierce the basement membranes, and come out of the circulation into the interstitial tissue and migrate to the point where they have to perform their function. Clear? This explains everything. Okay, and what stimulates them? These are all the stimulating agents: chemokines, microbes, cytokines. I have just told you all this. All right. Now comes leukocyte. Rolling to hoye gelo bhai, lege gelo, leukocyte and endothelium er moddhe attachment hoye gelo. Now the leukocyte has to exit the circulation. In order to do that, they have to migrate through the circulation, and this process is known as the migration of leukocytes from the endothelium into the interstitial tissue is known as what? Is known as, প্রথম শব্দটা কি লেখা আছে? Diapedesis. আমি ওই লাইন লিখবো না ভাই, I'm telling you, my method of teaching is not a cut copy paste. I just put a line and explain, you have to read. I don't put so much of effort. মানে আমি শর্টকাট নিই, I take মানে backdoor entry নিই, শর্টকাট নিই, আমি পড়ে দেবো. I'll explain, I'll just put the word, I'm going to explain, I'll put the diagram, I'm going to explain, you have to go and read. Reading is up to you. My job is to explain, your job is to read. Okay, so leukocyte emigrate ki kore korbe? The process is known as diapedesis, and this diapedesis mostly happens in post capillary venules, right? Again, adhesion molecules like PCAM, CD31 এগুলো আমি মোটামুটি বলে দিয়েছি. Leukocyte emigrate করতে কারা হেল্প করবে? In the interendothelial spaces we have these molecules like PCAM or CD31, junctional molecules. With their help, they come out of the circulation. But before coming out of the circulation, one more thing the leukocytes need to do. Interendothelial spaces with the help of CD31 and PCAM, they have entered the interendothelial space. But before they actually exit the circulation, there is one more function the leukocytes has to do. There's one more function the leukocytes must perform to exit the circulation. That is, it must pierce through the basement membrane. Basement membrane ke pierce korbe? Who, huh? Matrix metalloproteinase means various enzymes. One of them is collagenases. So these collagenases, they pierce the basement membrane and the leukocytes come out from the circulation. The leukocytes come out from the circulation. ঠিক আছে? So now the leukocytes have left the blood vessels rather, and they have entered the circulation. চলো ভাই, আমি কোথাও বেড়াতে গেলাম, আমি ল্যান্ড করলাম এয়ারপোর্টে, but that's not enough. Now I need to go to my hotel. Right? So leukocytes have to go to their, আমি একটা বেড়াতে যাইনি, চলো আমি একটা কাজ করতে গেছি, আমি একটা কনফারেন্সে গেছি. So I'm a speaker and I have been to a place where I'm going to deliver a lecture. আমি দিল্লি গেলাম, দিল্লিতে ল্যান্ড করলাম. এবার ফার্স্ট এন্ড ফরমোস্ট ইজ আমি হয়তো হোটেলে গেলাম, forget it. Now that's not final. I have to go to my place where I'm going to go to that hotel or go to that seminar place where I'm going to deliver the lecture. Leukocytes এর ফাংশনটাও সেই রকম, ব্লাড ভেসেল থেকে ইন্টারস্টিশিয়াল টিস্যুতে তো চলে এলো, কিন্তু that is not enough. Now it must go towards the point where it has to perform the function, right? So basically, the leukocytes will be going to the site of inflammation. Diapedesis to by blood vessel থেকে interstitial tissue, that is diapedesis. তারপরে কি লিউকোসাইটটা যখন interstitial tissue তে বেরিয়ে, when it has to go to the point where it has to fight the microbes, what is that process called? What is that process called? What is that process called? এত easy. Yes, what is it called? Not diapedesis. Diapedesis is emigration from the circulation, but now the leukocyte has to move towards that gradient. What is the process called? Huh? Chemotaxis. That is called chemotaxis of leukocytes. So there are some interleukins which are chemotactic. Any cytokines which act as a chemotactic agent is known as a chemoattractant. So one of the most important cytokines which act as a chemoattractant, known as chemokines, is interleukin 8. Interleukin 8. Interleukin 8 যখন secrete হয় না, this interleukin 8 attracts the leukocytes. After the leukocytes has come out of the circulation, after the leukocyte has come out of the circulation and now into the interstitial tissue, these chemoattractants will attract the leukocytes to the site of inflammation. So the most important chemoattractant is a cytokine known as interleukin 8. Interleukin 8, and this particular type of cytokines which act as chemoattractants are known as chemokines. They are known as chemokines, and the most important chemokine is interleukin 8. The most important chemokine is interleukin 8. Now the leukocytes will move towards the site of infection to perform its function. About in acute inflammation, which cells predominate in acute inflammation? My next question is, which cells predominate in acute inflammation? Neutrophils. Why? তোমায় জিজ্ঞেস করতেই পারে এক্সামিনার, আচ্ছা অ্যাকিউট ইনফ্লামেশনে কেন নিউট্রোফিলস আসে? হ্যাঁ, মোস্ট অ্যাবান্ডেন্ট, ভেরি গুড। তারপরে ভেরি গুড, এটা কি জানতে নাকি গেস করেছো নাকি পড়েছো? অ্যাবান্ডেন্ট, সেই তুলনায় গেস করেছো, ভেরি গুড গেস, পারফেক্ট। নেক্সট, ভেরি গুড আনসার। নেক্সট, কেন নিউট্রোফিলস আসে? ঠিক আছে, একটা হয়ে গেল অ্যাবান্ডেন্ট। ব্লাডে নিউট্রোফিলস কাউন্ট ম্যাক্সিমাম থাকে নর্মালি, তা নিউট্রোফিলস একটা, she said a very good point, নিউট্রোফিলস because of its abundance, নাম্বার বেশি। নেক্সট, হ্যাঁ, আমাদের আজকে আইপিএল দেখো, এত রেভিনিউ কেন জেনারেট হয়? আইপিএল এ এত রেভিনিউ কেন জেনারেট হয়? বিকজ অফ দ্য ভিউয়ার্স, because of the viewers. So নিউট্রোফিলের ব্যাপারটা এরকম, বিকজ অ্যাবান্ডেন্স বলে ওরা ম্যাক্সিমাম চলে আসতে পারে, এক্সেপশনস আর অলওয়েজ দেয়ার। যেরকম আমাদের আইপিএল টপ মোস্টে নয় এখনো, আরো লিগস আছে লাইক ইংলিশ প্রিমিয়ার লিগ, ন্যাশনাল ফুটবল লিগ আমেরিকার, ওগুলো আরো টপে আছে, কিন্তু আইপিএল যে রেটে উঠছে করে দিতে পারে। সো দ্যাটস নট দ্য পয়েন্ট, জাস্ট একটা এক্সাম্পল, আমি একটা কম্পারিজন করলাম। আইপিএল কেন এই অল্প টাইমে এত রেভিনিউ জেনারেট করছে? বিকজ অফ দ্য পপুলেশন, ইন্ডিয়ান পপুলেশন, আর এত লোক দেখছে। আমাদের নিউট্রোফিলসও কেন acute inflammation এ সবার আগে পৌঁছে যায়? because of the number, just the population এর জন্য, right? That is one point, abundance, population. Next, হ্যাঁ, ম্যাক্রোফাজ, নিউট্রোফিল ম্যাক্রোফাজ তো আলাদা। ওকে, আমি বলে দিচ্ছি, এটা একটু টাফ হবে তোমাদের জন্য। অ্যাবান্ডেন্স একটা চলে গেল, পপুলেশন। নাম্বার 2 ইজ এই যে অ্যাডহেশন হচ্ছে না, নিউট্রোফিলস সবচেয়ে ভালো পারফর্ম করে। They get adhered to the endothelium more avidly than any other leukocytes. That is why neutrophils predominate. In the first 24 hours, the neutrophils predominate. But after that, after 36 to 48 hours, neutrophils fade, they perform the function, they die, and the inflammatory infiltrate is taken over by chronic inflammatory cells like lymphocytes, macrophages, etc. Isn't it? But there are exceptions. There are exceptions. In Pseudomonas infection, neutrophils predominate. In some kind of viral infections, lymphocytes predominate. In allergic reactions, instead of neutrophils, eosinophils predominate. Okay, এই পিকচারটা জাস্ট এটা এক্সপ্লেন করছে, এখানে পুরোপুরি ক্লিয়ার কাট নয়. দিস ইজ নিউট্রোফিল. দেখো ফার্স্টে আসে নিউট্রোফিলস, and neutrophils are short lived. এই গ্রাফটা যদি দেখো, this is the neutrophils. First 48 hours এ neutrophils থাকে, they are short lived, and after that monocytes and macrophages, they stay for a longer period. That's why it's chronic inflammation. অনেক দিন ধরে ওরা থাকে. ঠিক আছে? So chemotaxis is done. So this is chemotaxis. Locomotion along a chemical gradient. Locomotion along a chemical gradient. So chemical gradient কি? Who does it? Interleukins. Especially which interleukin? It's a cytokine. What kind of a cytokine is it? Interleukin 8. So interleukin 8 is a chemoattractant. Chemoattractant. So chemoattractant cytokines কে আমরা কি বলবো? Chemoattractant cytokines কে আমরা interleukin 2, interleukin 4, TNF এগুলোকে কিন্তু আমরা ওই পার্টিকুলার নামটা নেব না. Any cytokine which acts as a chemoattractant is known as what? Chemokines. A perfect example of a chemokine is interleukin 8. Remember these points, write them and highlight them. Okay. So chemical gradients gulo ki, what are these chemical gradients or chemoattractants? These are the various chemoattractants. Okay, not only chemokines, chemokines is one of them. Excuse me, but there are other bacterial products, components of complement, cytokines like chemokines, arachidonic acid metabolism. ঠিক আছে? So this is over. Next is the recognition of microbes and dead tissues. Chemotaxis হয়ে গেল. এবার the neutrophils have reached the site. War zone এ পৌঁছে গেছে neutrophils, or the leukocytes have reached the war zone. ঠিক আছে? এবার war zone এ গিয়ে they have to kill the microbes. They have to identify the microbes. So what are the receptors? Toll-like receptors. Toll-like receptors. G protein coupled receptors. Just remember the names. Receptors for opsonin. Receptors for cytokines. These are the various receptors. These are present on leukocytes. And these receptors are specific for bacterial lipopolysaccharides. Bacteria এর মধ্যে lipopolysaccharides আছে, and these receptors are specific for bacterial lipopolysaccharides, proteoglycans, lipids, double-stranded RNA. Double-stranded RNA is present in some viruses, CpG nucleotides, etc., etc. So toll-like receptors which are present on leukocytes, they sense microbial products. So which products do they sense? They sense bacterial lipopolysaccharides, proteoglycans, and methylated CpG double-stranded RNA which is present in some viruses. Clear? Receptors are like, say, army men commandos who are identifying the terrorists. কি করে আইডেন্টিফাই করবে? There are certain particular features যা দেখে ওরা আইডেন্টিফাই করে. এখানেও এই পার্টিকুলার ফিচার্স কিছু আছে, receptors এই targeted particular features গুলো. What are the features? Like bacterial lipopolysaccharides, proteoglycans, DNA, RNA, CpG, etc. এই particular products গুলো দেখে, when this particular product is sensed, the toll-like receptors are activated. এমনি করবে না, সেখানেও কিন্তু অ্যাটাচ করতে হবে আগে. The leukocytes must attach to the bacteria, or they must identify the microbes they have to kill. Next one is G protein coupled receptors. G protein coupled receptors are mostly present in neutrophils and macrophages. What do they target? Short bacterial peptides containing N-formyl methionine residues. Amar mukhe ashe na sob shomoy. Even you don't need to remember all those. Just remember the names. Eta tomader easy howar jonno korechi. Then you have receptors for opsonins. What is opsonin? What is opsonin? What is opsonization? Opsonization কথাটার মানে কি? If a bacteria is targeted, what some of the leukocytes do is they cover the microbes get covered by particular substance. That is known as opsonins. And this opsonins এর মধ্যে receptors আছে, and leukocytes bind to the receptors and engulf the bacteria and kill it, engulf the microbe and kill it. So that process is known as opsonization. That particular process is known as opsonization. ঠিক আছে? You will come to know about it. হয়তো আজকে একটু বেশি বলে ফেলছি আমি. It may become a bit tough for you. So remember opsonins and opsonization. Okay, they are basically covered. They get covered by substances like opsonins, and the bacteria are targeted by neutrophils or eosinophils or whatever it may be, whichever leukocytes it may be, and they engulf the bacteria and kill it. That is known as opsonization. And finally, receptors for cytokines. So remember the name of these receptors: toll-like receptors, G protein coupled receptors, receptors for opsonins, and receptors for cytokines. Okay, তাহলে আমাদের কি এসে গেল? Recognition of microbes and dead tissues. ভাই আমি সবকিছু করলাম, লিউকোসাইট সার্কুলেশন থেকে বাইরে এলো, লিউকোসাইট সার্কুলেশনে আসে, সার্কুলেশন থেকে ইন্টারস্টিশিয়াল টিস্যুতে এলো, ইন্টারস্টিশিয়াল টিস্যুতে আসার পর কি হলো? It is moving towards the chemotactic gradient. Which is the chemotactic gradient? The microbes and dead tissues. After reaching the site of infection, they identify who they has to fight against. Recognition of microbes and dead tissues, identify korlo, and the final thing is this: killing and degradation. Killing and degradation. এটা বেশি টাফ হয়ে যাচ্ছে না? এটা বেশি হয়ে যাচ্ছে. Just remember the names. ঠিক আছে, you don't have to. I'm just explaining. I told you, no. I don't put points. I just explain on the basis of diagrams. আমি খুব কম পয়েন্ট লিখি. So if you take my PowerPoints also, if you listen to my class, it will be okay. Otherwise, you have to read and correlate. Okay. So the final one is recognition and attachment. Identify to করে দিল. এবার ওই যে receptor যেটা আমি বললাম একটু আগে, the receptors are through the ওরা attach করছে to the microbes. The microbes are getting attached with the leukocytes. Then engulfment. Engulfment কি হচ্ছে? This phagocyte membranes zips around the microbe. The phagocyte membranes zip around the microbe, and the microbe is ingested in a এটাই last. So basically a phagosome is formed. Okay, basically a phagosome is formed, and the microbe is inside the phagosome now. The microbe is inside the phagosome. Then the lysosomal enzymes, the phagosome fuses with the lysosome to form a phagolysosome. The phagosome fuses with a lysosome to form a phagolysosome. Phagolysosome হয়ে গেল মানে now it's ready. এটা গেল recognition and attachment. Microbes টা attach করলো with the phagocyte receptor. The phagocyte receptor zips around the microbe to form a phagosome. This phagosome fuses with a lysosome to form a phagolysosome. কেন লাইসোজোমের সঙ্গে অ্যাটাচ করছে? Why not any other thing? Because lysosomal enzymes will kill them. Lysosomal enzymes are right? The lysosomal enzymes will kill them. So a phagolysosome is formed. This phagolysosome, তারপরে কি লেখা আছে? Killing of microbes by lysosomal enzymes in phagolysosome. এবার কিলিংটা কি হচ্ছে, কিলিং এর প্রসেসে আমি আর যাচ্ছি না, কারণ I have already explained it to you. Free radical classes, a reactive oxygen species, reactive nitrogen species. It's going to take up a long time if I have to explain that. So I'm not getting into the details of it, neither they will ask you this question. এই মেকানিজমটা না, বাট কিলিং এই যে কি মেকানিজমে হচ্ছে এটা কিন্তু তোমাদের জানতে হবে। The microbes getting attached to the phagocyte receptors, the phagocyte receptor zips around the microbes forming a phagosome. This phagosome fuses with the lysosomes. Lysosome এর মধ্যে কি আছে? Lysosomal enzymes. And the bacteria inside the phagolysosome is killed, is degraded with the help of lysosomal enzymes. সেখানে free radicals এর ক্লাসে এক্সপ্লেইন করেছি আমি reactive oxygen species, reactive nitrogen species এটা। Okay?